Chapter 2, Cells, the Living Units, Part 1. So, the cell is the smallest living structure. Anything that is smaller than a cell is not considered alive. This includes the very structures inside of the cell and things like viruses. Viruses are not considered alive because they are not made of one cell. And of course, we humans are made up of trillions of cells. Cells need to perform certain functions to stay alive. The most important function, of course, is maintaining itself, maintaining the structures and doing the functions to stay alive. This includes, of course, acquiring nutrients, bringing nutrients and fluids into the cell. These nutrients are then used as building blocks to maintain the various structures found inside of the cell. And then, of course, any wastes that are produced need to be disposed of, returned to the outside of the cell. The other important function for many cells is replication, producing new cells. Cells come from other cells. And so that is necessary in the case of us humans in order to, say, repair damage to our bodies in some way, or say, to grow from being a tiny little baby to a full-sized adult. So the cell has three main regions. The most superficial region is the plasma membrane. The plasma membrane is the structure that goes completely around the outside of the cell. And so the plasma membrane is separating the inside of the cell from the outside. Then we have the large structure here called the nucleus. Nucleus is the largest organelle in the cell, and it holds the DNA. It houses the genetic information that allows the cell to perform its functions and makes it distinct from other organisms. And then we have the cytoplasm. The cytoplasm is everything found between the plasma membrane and the nucleus. So all the various organelles that we see in this cartoon, as well as the fluid that is surrounding those organelles. So the plasma membrane. Plasma membrane, as I said, separates the inside from the outside. The inside of the cell, of course, is the cytoplasm. The cytoplasm has the fluid called the cytosol. That fluid is aqueous-based. It is water-based fluid. So inside the cell, the fluid is mostly water. And then outside of the cell is the extracellular fluid. The extracellular fluid outside of the cell is also aqueous. It is also water-based. So the fluids inside the cell and outside the cell, while different, are both mostly composed of water. The plasma membrane itself is made up of structures called phospholipids. So each one of these little balls with the two little strings attached to them, each one of those is a phospholipid. And gazillions of phospholipids make up the plasma membrane. So here is a close-up of a phospholipid. So the spherical part of the phospholipid, that is a charged, so a positively charged hydrophilic phosphate group. Phosphate being a specific compound in the periodic table. Hydrophilic meaning water-loving. So the charged hydrophilic phosphate group likes to be exposed to water. And then the two strings coming off of the sphere represent the nonpolar hydrophobic fatty acid chains. Fatty acid chains are long chains of carbons and hydrogens, and they are nonpolar, so they have no charge, and they're hydrophobic, water-fearing. So the fatty acid chains do not want to be exposed to water. What this means is that the phospholipids will then arrange themselves into what's referred to as a lipid bilayer. As you can see, there are two sheets of phospholipids. In each sheet, the head, the polar head, the phosphate group, is exposed to the surrounding liquid, while the nonpolar fatty acid chains are inside the two layers, away from the water. So again, phosphates group is exposed to the aqueous solution inside the cell, and the fatty acid chains stay internal, away from the water. And the phospholipid bilayer is important because it acts as a barrier. it allows only some things to pass through. So certain materials can cross the plasma membrane, other materials cannot. Now, there are also other structures associated with the plasma membrane. This includes the proteins represented here by these big purple blobs. The proteins can be integral proteins. Integral proteins are embedded within the plasma membrane, so they may go partially into the plasma membrane, they may go completely through it. And some of these integral proteins, the one that go completely through the plasma membrane, are also known as transmembrane proteins. So transmembrane proteins are integral proteins that go from one side of the plasma membrane to the other side, so exposed to both the extracellular fluid and the fluid in the cytoplasm. Other big category of proteins are the peripheral proteins. Here's a peripheral protein, there's a peripheral protein. These proteins are not embedded into the plasma membrane. Instead, they lie on its surface. So peripheral membranes touch to some degree the plasma membrane, but do not go into it. We also have some glyco compounds. Glyco means carbohydrates. So we have some glycolipids. These are fatty acid chains attached to carbohydrates. So a glycolipid, fatty acid chains, the lipids attached to carbohydrates, the glycolipid. We also have glycoproteins. Glycoproteins are proteins that have carbohydrates attached to them. So glyco for the carbohydrates, protein for the protein. It's worth noting that these carbohydrates are always on the outer surface of the plasma membrane. So they are out facing the extracellular fluid. Turns out this is important because they allow our cells to recognize each other. Another compound found in the plasma membrane is cholesterol. Cholesterol is a lipid. Cholesterol is found in our plasma membranes, and they are important because they help increase the rigidity of the plasma membrane. So if you have the right ideal amount of cholesterol, then the cell's plasma membrane maintain the proper rigidity and are able to function properly. So again, the plasma membrane is a protective barrier limiting what can go in and what can go out. The plasma membrane also has receptors. Receptors are proteins that are associated with the plasma membrane. Often these receptors are transmembrane proteins, and they allow things in the internal or external environment of the cell to bind to the receptor, and this can lead to changes in the cell. So for instance, hormones released by other structures can bind to receptors in the plasma membrane of specific cells, causing those cells to respond in some way. The other important function of the plasma membrane is, as I mentioned earlier, its selective permeability. It allows some things to pass through the plasma membrane, so small nonpolar molecules can enter or leave the cell through the plasma membrane, and also water. Water can go in or out of the cell freely. Other things, large molecules, large structures, and things that are charged cannot pass through the plasma membrane. They are blocked from going into or out of the cell. All right, so things moving across the plasma membrane. They do this by a variety of methods. First method being passive transport. Passive transport where materials move along their concentration gradient, so going from where there's a high amount of them to where there's a low amount, and it does not require any energy from the cell, which is awesome. It means it's free. The cell doesn't have to work to get things to move from high concentration to low. One common form of passive transport is diffusion. So for instance, here we drop some dye into this water in a beaker, high concentration of dye in the droplet, low concentration in the water, and over time the dye molecules will move from high concentration to low until we get an even distribution of dye. For our cells, the material is going from high concentration to low, crossing the plasma membrane. So simple diffusion, lipid-soluble solutes, nice and small, can cross the plasma membrane going from high concentration to low. Water molecules do the exact same thing, going from high concentration to low, And that process is called osmosis. So osmosis is the simple diffusion of water molecules. Now it turns out that there are ways for materials that are blocked from passing through the plasma membrane to actually be able to cross. And this requires transmembrane proteins. So solutes, ions, things that cannot normally pass through the plasma membrane can go from high concentration to low by passing through very specific transmembrane proteins. So the diffusion is facilitated by these special proteins that act like windows to allow the molecules to enter or leave the cell. Then we have active transportation. Active transport involves things moving against the concentration gradient. So going from low concentration to high, going from there's very little of it to where there's a lot of it. And this, since it's going against the gradient, requires energy. The cell must expend energy to engage in active transport, and that energy is in the form of ATP, adenine triphosphate. The energy currency of the cell is ATP. So here's an example of active transport. Here we have a special protein in the plasma membrane, and it will move something from low concentration to high. Maybe this is a waste the cell wants to get rid of. Maybe it's a nutrient the cell wants to store. And the cell uses ATP, uses that energy to cause the molecule to get pumped from one side of the plasma membrane to the other. So transmembrane proteins pumping something from low to high, from low concentration to high concentration, using energy to do it. Materials can also move into or out of the cell using vesicles. Vesicles are spheroids made up of the phospholipids. So these are very similar structurally to the plasma membrane. And they can bring things in via endocytosis. So say a large food particle, the plasma membrane can form pseudopods surround that particle. Then the pseudopods meet, pinch off, forming a vesicle inside of the cell, holding that food particle. So endocytosis, bringing it into the cell. Exocytosis is the opposite. A vesicle is formed inside of the cell, filled with something the cell wants to release. The vesicle then travels to the plasma membrane, fuses with the plasma membrane, and releases its contents outside of the cell. So exocytosis lets things exit the cell. Now, nowhere in here do we see ATP being used, but it turns out a lot of ATP is required for changing the shape of the plasma membrane and for moving vesicles into or toward the surface of the cell. Two types of endocytosis. We have phagocytosis. This involves bringing in a large solid structure. So this is cellular eating. Phagocytosis, bringing something big and solid into the cell. The other type, so here's an example of phagocytosis where a white blood cell is surrounding, bringing in a microbe with the intent being to destroy that microbe. So our white blood cells use phagocytosis to bring in pathogens, microbes, and destroy them. The other form of endocytosis is receptor-mediated endocytosis. Here, molecules bind to protein receptors, so proteins in the plasma membrane. When the molecules bind to the receptors and enough of the receptors are filled, then the receptors will cause signal for the vesicle to form, bringing that very precious material into the cell. So receptor-mediated endocytosis. Unfortunately for us, viruses can sometimes take advantage of receptor-mediated endocytosis. The viruses will have on their surface little proteins that will mimic the substrate and bind to those receptors, tricking the cell to bring the virus into the cell. So example, influenza uses this approach to get into our cells and then take over the cell, turning it into a virus factory. So here, viruses at the surface of the cell, virus gets brought in, and then now it's inside of the cell. And this is where we'll stop for this part of the lecture.